conversations. Thanks. These events are organized, coordinated, and facilitated by staff from multiple agencies. And I want to thank the TPCC planning team and 1850, which is the coastal program, 1854 Treaty Authority, and Minnesota Sea Grant. And I want to also note that funding for Minnesota's coastal program is provided by the National Oceanic and Atmospheric Administration Office for Coastal Management. If you're new to the Twin Ports Climate Conversations, this is a program that's been around since 2016. We offer quarterly conversations and generally offering a space to gather and discuss implications of a changing climate in the Western Lake Superior region. These events do cover many different topics. We focus on perspectives of climate change impacts, adaptation responses, and opportunities for mitigation and resilience. You are invited to join the conversation as well as the production of these events. Please do reach out if you're interested in helping support these events. And with that, I'm going to hand it over to Steve to introduce himself and give a little more information.
Yeah, thanks, Julie. Hi, everybody. My name is Steve Shire. I am the Natural Resource Specialist for the 1854 Treaty Authority. If you're not quite sure what the 1854 Treaty Authority is, we're an intertribal organization. We work for Bois Forte and Grand Portage bands of Chippewa here in Northeast Minnesota, and we work to protect and preserve their off-reservation treaty rights. I just want to thank everybody for joining our webinar today. Just a quick reminder that we are using that Q&A function, and we will wait to answer any questions until the end. If you'd like to learn more about our program or see any of our previous webinars, we do have a website, which will be linked in the chat. Additionally, there is a link there to subscribe to our email list for upcoming events. So at this time, I think we just kick it off. I'd like to introduce our three panelists for today. First off, Leanne Sethna. She's an assistant scientist at the St. Croix Watershed Research Station. Sam Blackburn, he is a data scientist at the University of Wisconsin-Madison for limnology. And Chris Philstrip, he's a limnologist at the University of Minnesota's Natural Resources Research Institute. So thank you so much to these three for taking the time to join us today. And I'll turn it over to Leanne.
Awesome. Thanks, Steve. Here we go. This is working. I'm going to be chatting today about a project we've been engaged in for the last couple of years, studying the formation of harmful algal blooms in lakes across the northern part of the state. And this research was funded by the Environment and Natural Resources Trust Fund, which is a state program that funds a lot of environment and natural resources research in the state of Minnesota. And as Steve mentioned, I am a research scientist at the Science Museum's research station, and a lot of our work has also been done in collaboration with 1854 Treaty Authority and the Red Lake DNR. Okay, so when we think of our precious northern Minnesota lakes, we often think of landscapes that look like this: beautiful, clear, blue water, and loons. But recently, there have been more and more reports of these harmful cyanobacterial blooms that are occurring in lakes within the boundary waters and within the Superior National Forest and other lakes that are considered to be within more pristine or undisturbed watersheds. And there have actually been a lot of studies that document harmful algal blooms in lakes that we don't primarily consider to be at risk for eutrophication or other kinds of nutrient impairments. And so these photos that you see, one's from Lake Superior shoreline that I believe Sam will touch on in his talk. And then these photos on the right here are photos that we took in Smoke Lake, which is only 2 or 3 portages into the boundary waters themselves. And so these blooms represent a significant concern because, one, they're used for backcountry recreation. So people who are out here on these lakes are relying on them as a source of drinking water. And it's also an ecological and environmental concern because so many trophic pyramids and the way nutrients and carbon is cycled in these lakes is disrupted when the plankton community shifts into domination by cyanobacteria. And harmful algal blooms are in the news. A lot of reports have come through talking about toxic algal blooms, especially in lakes where we wouldn't necessarily think of being at risk for harmful algal blooms and the toxins that they produce. And so this prompted a first round of study within the Superior National Forest and within the Boundary Waters Canoe Area Wilderness. We engaged in a full year of water quality monitoring where we were taking samples to measure the nutrient concentrations and the phytoplankton community. And we also deployed these high resolution buoys that were able to collect dissolved oxygen and temperature data from each of our 8 study lakes. And these buoys gave us some really great data because initially, a lot of our study lakes were relatively shallow. You can see that they range between like 4 and 11 meters deep. So that's about 15 to 30 feet. And so we expected a lot of these lakes to actually be what we consider well mixed, meaning their temperature profiles are the same from the top to the bottom. But what these buoys were able to show us is that we actually had a range in the ways in which lakes were stratifying and mixing during this summer period. So you can see that we had 2 lakes that we consider to be well mixed, meaning their temperatures are the same from the top to the bottom. So these black lines are connecting areas of equal temperature, and the vertical lines indicate that the temperature is the same all the way through the water column. We also had lakes that are called dimictic, meaning they are thermally stratified during the summer and they mix fully in the spring and the fall. And so these are the lakes that you can think of when you're fishing or when you're swimming and you find areas that are much colder at the bottom, or when you're swimming and you dive down and you can feel that colder water. So those are lakes that are thermally stratified, and it forms kind of a barrier between the bottom and the top. And then we had these lakes that were right in the middle. They were thermally stratified for some of the summer period, but they also mixed multiple times within that monitoring zone. And we also found, this was significant, because these lakes that intermingled between being stratified and being mixed were also the lakes that were experiencing harmful algal blooms. And the timing of these blooms aligned to when lakes would be stratified and then mix. And then after this mixing period is when we observed the formation of cyanobacterial blooms. So it led us to ask this question: how does mixing relate to the formation of harmful algal blooms? And our hypothesis is that it's facilitated by this mechanism known as internal phosphorus loading, which works because lakes, when they're oxygenated and well mixed or oxic throughout the entire water column, phosphorus doesn't really come out of the sediments. Phosphorus is this naturally occurring nutrient, and so we find it in lakes, and it does facilitate algal growth. But when lakes are oxygenated, there's little phosphorus coming out of the sediments. But when a lake stratifies, it creates this barrier between the bottom waters and the surface waters, and the bottom tends to go anoxic or lose all of its oxygen. And so under these chemical conditions, phosphorus actually comes out of the sediments at much higher rates. And so there's a lot more phosphorus being loaded into these bottom waters. And then when the lake mixes, all of the phosphorus that's contained within the bottom waters is circulated up through the water column and becomes available for algal growth. And so this process continues in all of these lakes that are called polymictic. And so they're going through these cycles of loading phosphorus into the bottom waters and mixing it into the entire water column multiple times in the summer. And so what we found was that these polymictic lakes are experiencing these cycles of anoxia and internal loading. And we're calling this process anoxymixis because the lakes have to go anoxic. You can see that in all of these graphs along the top of the chart here, the red lines go down towards zero multiple times in the summer. The blue line indicates how stable the thermal profile is. So when the lake is stratified, the blue line is high, and when the lake is mixing, the blue line is low. And you can see that for all of these lakes, there's this flip-flop that happens. So the lake stratifies, the dissolved oxygen goes down, and then you get mixing. So the lake mixes and dissolved oxygen is circulated throughout the entire water column. And so all of our lakes that had these anoxymictic regimes and anoxymictic processes were lakes that facilitated cyanobacterial harmful algal blooms, or cyano-HABs. And so as we think about the ways in which climate is affecting the ways in which lakes stratify and mix and go anoxic, there's a lot of literature that suggests that these shallow lakes are going to be more prone to this process of anoxymixis. So as we warm our surface waters and reduce the amount of oxygen in the bottom waters, we're setting up lakes to be more at risk for internal loading and for cyanobacterial harmful algal bloom formation. And so this has prompted us to start a new study in which we've worked closely with Steve and his team at 1854 and a team of scientists at Red Lake DNR to study lakes across a broader geographic region within northern Minnesota. So we're monitoring lakes both within the Red Lake area and within the 1854 ceded territory. And some of those lakes are going to fall again within the Superior National Forest. And we've targeted lakes that are primarily within forested and undisturbed watersheds. And they span this gradient of lake depth and surface area. And so we are again engaging in water quality monitoring and buoy deployment that can help us understand what lakes are going through this anoxymictic process and how that affects surface water chemistry and algal ecology. We're also going to be taking sediment cores from lakes in order to recreate the history of algal ecology and nutrient availability and ask questions like, have lakes always been supporting cyanobacterial blooms? Or is this a trend that aligns with changes that we know are occurring due to climate in Minnesota? And we're taking explicit measurements of how much phosphorus is actually being loaded from the sediments using incubation experiments. So these are all cores we actually just took from 3 lakes within the Superior National Forest, and they're currently in our incubation fridge in the lab going through these cycles of oxic conditions, so oxygenated conditions, and non-oxygenated conditions. And we measure the overlying water to calculate how much phosphorus and nitrogen is coming out under these 2 treatments. And so this will be how we explicitly test whether internal nutrient loading is contributing to the excess nutrients and the cyanobacterial blooms. And so using this combination of monitoring, paleolimnology, and lab experiments, we hope to be able to better describe the gradient that is controlling lake water quality and algal blooms across these relatively pristine and undisturbed lakes in northern Minnesota. And so with that, I'd like to thank some members of my team that have contributed a lot to the fieldwork and the analyses. And again, thanks to my project partners at 1854 and Red Lake and the LCCMAR.
Awesome. Thank you, Leanne. Appreciate the overview on what you're working on. Next up, we have Sam Blackburn.
Awesome. Let me just get everything running now. All right. There we go, folks. So I'm going to be talking about a bigger lake than Leah did, but I'm going to be talking about a lot of similar themes, talking about water warming and phosphorus, and hopefully you'll see some through lines between what can cause cyanobacterial blooms in both an extremely large lake and the smaller lakes in northern Minnesota. So as you may have noticed, if you saw the listing for this event, I am not Dr. Kate Reinl. I am filling in. She is here to answer questions, but uh, I'm someone who works with Kate a lot, and we'll be filling in and talking about cyanobacterial blooms in Lake Superior. So without further ado, I'm going to get started. So as Leah mentioned, we expect northern Minnesota and Lake Superior to be relatively pristine. It is probably we think of it as the most pristine Great Lake. Because compared to something like Lake Erie, which we associate with having algal blooms. And that's not to say that there aren't we don't see water quality changes in Lake Superior. In fact, we often see sediment plumes following extreme storms. In fact, huge sediment plumes that you can see from space. But we don't expect to see things like cyanobacterial blooms. And yet, these have been happening more frequently in the Great Lakes, and particularly in Lake Superior in recent years. Before, I just want to define how I'm going to be referring to a cyanobacterial bloom. It's any aggregation of cyanobacterial biomass as opposed to some other algae in the water in some part of the water column, which may lead to a surface scum or may lead to some sort of buildup further below the surface. So the key thing here is that we're going to be talking about cyanobacteria as opposed to some other algal species forming a bloom. And also, I just wanted to mention that cyanobacterial blooms in Lake Superior can look really different. You can have these really bright paint scums, or you can have more of a deeper water being tinted green. So if you look at what is a cyanobacterial bloom, it may not always look like what you expect to see if you're used to seeing it in smaller lakes, which often have the more well-defined paint scum, like you can see on the further right of my screen here. So if you're new to hearing about cyano blooms in Lake Superior, I just want to give a quick rundown. A lot of this is based on the Lake Superior Bloom Bulletin that the Lake Superior National Estuarine Research Reserve puts out. I can throw a link to that in the chat after I speak, but just to give you a sense of what's going on and why there's some concern here is there's been reports of blooms in numerous parts around the western half of the lake, as far up as Thunder Bay. There have been numerous reports in the St. Louis River estuary, as well as a hotspot around Moakaway and Siskiwit Bays near the Apostle Islands. And reports do seem to be becoming more frequent. We are seeing have been getting reports of blooms more frequently since the first noted bloom in 2012. That's not to say that they haven't been occurring before now. It's just that with increased attention, we do seem to be noticing increases in the number of blooms along the lakeshore. And you also tend to see blooms later in the summer when you have warmer lake waters. And as you can see, so August and September tend to be when we see most of our blooms. And I do also want to note that we refer to these often as harmful algal blooms, but in a lot of cases, the blooms detected along the shore of Lake Superior proper have not been toxic. So there have not been levels of microcystin above the EPA advisory limit. In fact, it's only been detected once in Lake Superior. There have been other toxins, so it's not to say these aren't harmful to the ecosystem at large or cause effects. It's more to say that the most common harmful impact to humans of algal blooms has not been present in Lake Superior at this time. So the reason, or one of the main things I have something to talk about here is that the Lake Superior algal bloom and nutrient subgroup, which is a part of the collaboration between the Canadian and US sides of the lake, a lot of different research organizations have come together to try and understand why we're seeing blooms happening in Lake Superior. And as you can see, there's a lot of logos here, a lot of different organizations, a lot of people to thank. I'll touch on some of those at the end of my talk, but really just want to give you the sense that this is a huge collaboration between a lot of people who care about what's going on in the lake. And the way that those efforts are coming together involves teaming up to collaborate on monitoring initiatives to cover more of the lake. My image here is a collaboration between the Wisconsin DNR and what is now the Burke Center for Knowledge. It used to be a part of Northland College, to regularly sample along the lake and look for changes in water quality, water temperature, detecting different potential cyanobacterial species in the water. In addition to collaboration on monitoring, you have shared knowledge between different groups. That's one thing that I'm part of why I joined the project, was to help increase the synthesis of data between the different organizations. So combining everyone's data sets into one larger data set that could be used for scientific research. And also, yeah, just shared reporting. So like the Bloom Bulletin is bringing together knowledge and reporting between different organizations to better communicate with the public. So I'm going to get a little bit into the science and touch on what we've learned from this collaboration between different organizations. So as a similar themes to what Leanne mentioned before, just want to I don't want to bury the lead here. Blooms are more likely with warmer lake temperatures and elevated lake phosphorus concentrations. And so some of the reasons that we have come to understand that, I want to start with one really sort of really focused study that Kate led with some others in the field, in the region, that really looked specifically at those two specific causes. So Kate went out and gathered sediment and gathered water from tributaries, the lake proper, and harbors, and then conducted incubations under different conditions to understand how different cyano and algal species grew. And what Kate and her co-authors found was that under warmer water conditions, so 20 degrees and 25 degrees Celsius, so that's, someone can throw in the chat, but it's 60, 70 degrees Fahrenheit, and under conditions where there's higher levels of phosphorus compared to nitrogen, which are the different elements, so higher levels of phosphorus, we saw the most blue-green algae growth. That's what I've highlighted in this red here. And also, they saw that growth most commonly in waters taken from rivers and harbors. And that's notable just as we talk about the influence of storms and flooding events as bringing in phosphorus, increasing phosphorus in the lake, which can lead to blooms. It also could be bringing cyanobacteria themselves, which then are able to bloom once they reach the lake. This yellow color is the species of cyanobacteria that has been most commonly associated with blooms in the near shore of Lake Superior so far. So talking a little bit more about temperature. This is a plot here of degree days, which is a measure of overall lake water warming over time. So 1 degree day is the sum of average daily temperatures in the lake. Basically, more degree days, the warmer the water has been over the course of a season. And you can see here, these years with lower degree days are the years where we've had very little, basically no bloom activity at all in the nearshore lake. And up here, this highest is 2012, where we had those first blooms, and 2024 was a fairly active bloom year as well. So you can see there's a clear distinction between colder water years without blooms, and then blooms sort of requiring a certain amount of water warming to occur in the lake. Which is relevant because Lake Superior is one of our most rapidly warming lakes. You can see over time, over the past 40 years, we have a significant increase in the maximum summer lake surface temperature. So this is something that could potentially make blooms more possible as time passes. The other side of things I want to talk about, as we saw, having higher concentrations of phosphorus in the lake makes blooms more possible and encourages bloom formation. One thing that leads to those conditions is floods, is storm events. This here is a plot sort of looking at generally, was there more water moving into the lake from tributaries? And this is looking at over the previous 2 months before the lake was measured, and you can see in periods where we had more flooding, more tributary discharge into the lake, we saw higher lake phosphorus concentrations. And also, if you're looking, and particularly looking at floods, 3 of those years with the most notable bloom activity, those being 2012, 2018, where it's had an enormous bloom along most of the lake's period near shore, near the possible islands. And then 2024, which was a fairly active bloom year as well, all had large floods that occurred in the early part of the summer in June, and that seems to have a clear connection with the more active bloom year following. And again, this is something of a concern because as the climate changes, we are expecting extreme flood events to occur more frequently, like those that led to those increased bloom years. As you can see, so those 2 of those peaks that we saw on the previous slide, 2012 and 2018, were 1,000-year floods that happened 6 years apart. These things are becoming more common, unfortunately. So jumping back to those main points, blooms are more likely with warmer lake temperatures, with more warming, and with elevated phosphorus concentrations, both of which are likely to occur more often in Lake Superior. And so it means blooms might become more likely over time. And so that's why it's important for us to understand more about when they occur and if there's anything we can do to reduce that likelihood. So lots of people to thank here. I want to specifically shout out that the funding for my part of the research comes from the University of Wisconsin Division of Extension and also the Office of the Vice Chancellor for Research. I want to shout out Brenda and Matt and Ellen in particular are members of the team that advises my work, and also just everyone on here has contributed to the greater knowledge of blooms in Lake Superior. So thanks to all that, and yeah.
Great, thanks, Sam. Appreciate you stepping in there last moment to be with us today. Let's see, finally we got Chris Philstrup, last but not least here, but just a reminder to everybody viewing that if you do have a question you want to answer, then feel free to put it in the Q&A section of the Zoom, which should be found on your lower bar there, Q&A with a question mark. So with that, I'll hand it off to Chris.
All right, thank you. Thank you everyone for being here. I will try to get my screen adjusted appropriately. And I think you are looking at what you should be looking at, so that's good. If not, someone just yell at me, but I'll go ahead and get started. Thank you all again. So what I'm going to be talking about today is a project that we're just starting now. This was a project that was funded during the 2024 round of the Environmental and Natural Resources Trust Fund, LCCMR process. So we put in this proposal a while ago, but the funding just started this July. So we're just getting started. I do want to note that my collaborator and actually the PI on this grant is Hillary Sorensen from Minnesota Sea Grant. Many of you probably know Hillary. So Hillary's on leave right now. She'll be back soon. So I put this together without any of her advice. So any mistakes in this presentation are solely my own. So it's just a little preface to get us started. But what we're going to be doing on this project is actually working with lake communities to track some of the bigger stressors that we're starting to think about with climate change and how we deal with climate. And most of these are focused on chloride and actually harmful algal blooms. I do want to say we are not saying there is a link between chloride or road salt and harmful algal blooms, but these are just 2 of the concerns that we've come across when we've talked to our communities up in northern Minnesota. All right. So I think most people have heard of chloride and why we're concerned about chloride or salt pollution of our fresh waters. The title of this slide is a little play that came out of a paper called Salting Our Fresh Waters, which looked at how there is kind of this national concern that we are seeing elevated salinity in waters across the United States. And how does this relate to the 14,380 Minnesota lakes we have? We just don't know that yet. So this is something we want to start monitoring. Salt pollution can come from several sources. It can come from road salt. It can come from water softeners, especially water softeners in rural areas where there may be a septic system or something like that that doesn't really treat that salinity. And also dust suppressants, which was kind of surprising to me because I live in the country on dirt roads, but dust suppressants can also increase salt concentration in waters. And Leanne did a really good job setting this up. I mean, the concern with salinity is it can cause lakes to permanently stratify, meaning they don't mix from top to bottom. And that could be a gradient that's not necessarily driven by temperature, but it could be driven by the density because salt water is denser than surface waters. And eventually we could get to a point where those waters are not mixing. That could lead to issues with, well, just higher elevated salinity in these lakes, which could have issues for fish and other aquatic organisms. It could lead to permanent stratification and building up of things like heavy metals, nutrients, and things like that in the bottom water, which potentially could start leaking into the surface waters. So this is what we're really concerned about. And if you've seen any of the reporting coming out of the Twin Cities, this is a real strong concern down there, but we don't really have a good handle on how this is impacting our rural ecosystems. I'm not going to talk much about cyanobacteria because I think we've covered it all pretty well already. But in addition to the toxins and kind of that public health concern, like Leanne was showing, I mean, there's also concerns about just deoxygenated bottom waters and not having available oxygen down there for organisms like fish and fish food organisms like macroinvertebrates. Again, this could lead to issues with internal loading, like Leanne was saying. So harmful algal blooms kind of have an impact on two things, the public health perspective, but also the suitability of ecosystem habitat. And so in the land of 14,380 lakes, how do we monitor these changes that we're seeing? How do we monitor everything everywhere all at once, to play on the popular book and movie title, when you have all these systems? And, when we think about our organizations and our agencies that have limited funding that they really need to target on the most pressing issues, how do we get this sense of how things are changing that aren't the most impaired? And I think our answer to this through this project and through other really great projects out there are by establishing this participatory science network. I love this image. I didn't cut any of it off because I wanted to show you how great it looks. But think about, we have a lot of people out there. And one of the things I still marvel at, I didn't grow up with a cell phone. I didn't have a cell phone until I was out of undergrad. But people now have basically the computing power that's superior to the original Apple I computer in their hand. So we have a lot of people that are out there, a lot of people that are concerned, and a lot of people that are thinking about ways that they can contribute to developing some of the solutions to these problems. And so that's really what we want to aim to do on this project. There's way too much text on this slide. I apologize. But basically, we want to work with lake communities to start monitoring chloride and toxic cyanobacterial blooms in the most rural lakes. So those lakes we think about in northern Minnesota. We're going to use this community-based participatory science approach to establish a volunteer network of people to help out with this. And we have several outcomes here. And the one I just want to highlight, because I don't have a good mock-up of this yet, but we will be working with these communities where they can take their data, upload it to a project management system, and then there's an interactive data portal that they can see past data that they've shared with us, and we can interact with these communities in addition to the time that we get to go interact with them in person. So that's kind of our approach, what we're thinking about. What this looks like is putting kind of tools in the hand of people to help us monitor, and then pairing those with laboratory-based measurements that might be a little more accurate. And we can start building relationships between the accuracy of tests that people are contributing to versus what we see in the lab, to look at what those relationships look like and get some better information off of these lower cost, maybe simpler techniques to monitor surface waters. And so we're going to do that with chloride. I should have said that. So some chloride monitoring. And this we kind of view as a stress that'll happen earlier in the season. So once we get like snow melt, the spring flushing, that's when we should see higher chloride measurements moving into these systems. We'll also work with cyanotoxin monitoring. For this project, we're going to just focus on one type of cyanotoxin called microcystin, which is, there's like 1,200 different types of microcystins, so we just call it microcystins. But we'll be giving kits to the volunteers. And if you read, there's a little QR code here to a report that Janae Whitaker, formerly of the Lake Superior National Estuarine Research Reserve, did a lot of research on the accuracy of these test strips. So if you want to go ahead and look at that report, that would be a great place to start. But we're going to compare this with some kind of the state-of-the-art laboratory techniques for improving accuracy of those concentrations. And this is my colorful schematic, the beta version, which means Hillary hasn't seen it. But basically, the project team is going to work with soil and water conservation districts and lake associations. So kind of our project partners to develop these approaches and training sessions and things like that. And from there, we're hoping our project partners will help us organize the actual volunteers. They'll help identify local volunteers. They will help us with training and distributing kits, and also help share findings with local volunteers. The local volunteers will be doing all of the hard work, including collecting samples, analyzing data, and uploading data to this portal. And I do want to say that we're going to start with a limited group to begin with, but this is a multi-year project. So we're going to learn a little bit during the first year, which is next year. And then we're going to expand our capacity into 2027 and bring on more groups and volunteers. So anyhow, sampling will begin next year. So we're excited about this. Like I said, Hillary will be coming back soon. And Hillary and I will be meeting later this fall and winter to start developing some of these protocols and then reaching out to our partners. This is all a huge plug just to say, if this is something you're interested in, reach out to your local soil and water conservation districts or other local organizations, or reach out to me and just let us know that you're interested. My email is right there. And with that, I will end so we have time for questions.
Awesome. Thanks, Chris. And it already looks like you might have one volunteer in the Q&A. Dan Ratliff looks like he's looking to do some volunteer work. So I don't know if you mind putting your contact information in the chat just as a reference. Dan, that might be a good spot for you. So again, I just want to open it up for questions at this point. Just remember to put your question in the Q&A function if you have one. I guess I'll start off here. Let's see. Mary Beth asks, Dr. Philstrip's bio cites research focused on intensively managed agricultural regions. Any advice, question mark? I would assume maybe any advice on the management of those agricultural regions?
Yeah, so this is where I have to admit I am not a native Minnesotan. So before I moved to Duluth, I was in the state of Iowa, working at Iowa State University. And before that, I was in the state of Texas, working on drinking water reservoirs and HABs down there. So I've kind of traveled up the I-35 corridor. But I would say, working with your local agricultural extension agents is probably the best place to start, either them or local soil and water conservation districts. There are a whole host of best management practices to try to reduce the amount of fertilizer running off of farm fields and agricultural fields. But they're not always, it's not a one size fits all approach. Depending on the systems and the challenges you're dealing with, you likely need to tailor what the combination of those best management practices look like. But basically, it's reducing fertilizer applications if not needed. It's keeping soil on the land, so cover crops and things like that, and reducing the rate at which water runs off the land and delivers those nutrients. Those are kind of the high points, and the different best management practices are built into that.
Great. Leanne, it looks like you had a comment about the Q&A question.
Yeah, I wanted to add too that we don't have a citizen science program as part of our project, but we are trying to collect data from people who have observed blooms in potentially unexpected places. And I have a Google form that takes in where and when people have seen these blooms, because as part of our project, we'd like to better refine what we know about lake shape and lake depth and how that correlates with bloom formation. I tried to put it in the chat, but it seems like it's disabled. So I'm not sure where to put the link, but I have a form. So if Dan, as part of your sampling on Chemo Lake, if you notice blooms, you could report that via the form, or anyone else who's observed blooms and wants to report them. We have a mechanism for that.
Great, perfect. Maybe we can figure out how to get that out to the people that registered. Oh yeah, send it to Julie. Perfect. Okay. Let's see, got another question here. Kind of just gonna open it up to anybody. John asks, is there a surface water temperature threshold that exacerbates or kicks off the seemingly annual algal blooms?
I don't know if that's going to Leanne or to me.
Yeah, I think I'm just open to whoever has an answer for it.
Can you?
Yeah, I mean, it varies a lot based on the size of the lake. We definitely don't have a threshold in Lake Superior. There's still so much we need to know. We just have a general sense that warmer makes it more likely. I'll let Leanne weigh in if there's anything she knows more about the smaller lakes.
Yeah, I think that actually a lot of Kate's work has shown that cyanobacterial blooms also bloom when water temperatures can be cooler. Cool thing about cyanobacteria is that they were like one of the first photosynthetic organisms on Earth, and so they're really like evolutionarily prepared to grow in all types of waters and conditions. Typically, the toxic blooms are most frequently observed in like the late summer when water temperatures are higher. When we noticed blooms, it was actually the end of September. And so you think that it's fall, the air temperature is cooler, but the water is still warm. It's had the whole summer to warm. So that's, yes, summer or early fall typically.
Let's see, got another question here. Dan asks, what characteristics of a lake make it more susceptible to blooms?
I'm sorry, I'm getting distracted by like the chat and the Q&A. Can you repeat the question?
What Dan asks, what characteristics of a lake can make it more susceptible to blooms?
So in our work, we're focusing on identifying lakes that have this mixing phenomenon occurring. And so we think that lakes that have a larger surface area and a shallower depth are more likely to be experiencing midsummer mixing. And so that's what's going to be kicking up these nutrients and then contributing to bloom formation.
Yeah, I would say the other thing is lakes that have a really big watershed that gets focused down into a smaller lake. Those have the capacity to absorb or take in a lot of nutrients from a really big area and funnel it down to a smaller thing. Lakes in developed watersheds tend to have issues with nutrient, especially phosphorus loading. Lakes in agricultural systems tend to have really high nitrogen and phosphorus loading. The thing is, those aren't the lakes we're studying up here, right? So lots of what we know about what causes cyanobacteria to form is not what we're seeing up here, which signifies that there's a change in these lakes due to thermal structuring, due to the frequency of these big storm events and pulsed events, loading nutrients like Sam showed. So there's still a whole lot we don't know, but yeah, I would say shallow lakes, lakes with big watersheds, developed watersheds, agricultural watersheds, all of these things kind of help contribute to that. Also, lakes that don't have many plants in them, aquatic plants. Aquatic plants tend to suck up and bind a lot of nutrients, so the algae can't get it. So that's another concern is if you don't have any aquatic plants sucking up those nutrients, those become available to algae and cyanobacteria and things like that.
Great. Got a couple more here. Glenn asks, in northeastern Minnesota lakes, is there a correlation between the likelihood of cyanobacteria blooms and shoreline development?
Yeah, I guess I could just jump in because that's a little similar to what I was talking about. And, I think it's variable. I'm trying to think if I can think of a paper that shows a really strong correlation, and I don't know that I do, because not only is it associated with the development, but it's also associated with like the amount of green infrastructure on those shorelines, which can help reduce nutrients flowing into the system. I've been at some lakes where you see row crop agriculture right up to the side of the lake, right up to the lake shore. I've also been in other systems where you see green lawns being fertilized right up to the shoreline. So these are things that could contribute to that. So those would be developed shorelines, but also just to put a plug in, there are some very easy best management practices that you can do to help reduce those concerns. And I was recently working with the Cook County SWCD on a workshop up there where we kind of ran through some of those. So again, plug your local SWCD, go chat with them. There's ways that you can help contribute to the solution to these algal blooms.
Kind of, I guess in that kind of same vein, what changes to management of our surface waters need to occur to protect them? From Nicola.
Well, I think that's a great question and also a complicated one because there are so many facets and the ways that we're modifying our landscapes, changing our climate, and those all contribute to the ways in which our water quality is degrading. A lot of our research in these relatively pristine systems point to more regional action. For example, warming. Warming is like a really big contributor to the formation of these blooms in our pristine lakes, and there's not any more watershed manipulation that can be done. It's already a forested, undisturbed landscape. And so like climate action is really important. We've also found that like these lakes are not so far gone. You know, like the paleolimnology data suggests that they're not completely different lakes. So it's not like we've entered a whole new like era of cyanobacterial blooms all the time everywhere. And so there's still like action that can be taken to reduce the risk of blooms into the future.
Yeah, I guess kind of also in the same vein, are there state or federal policy solutions here? How does the Clean Water Act apply?
Yeah, I mean, Leanne, if you want to keep going, I mean, I think this,
Yeah.
kind of leads to this complexity of jurisdictions and what different groups have the capabilities to handle and coordinating and working together. So yeah, go ahead, Leanne.
Well, the Clean Water Act just sparked something in my brain. There's a lot of exceptions within the Clean Water Act, like for non-point source pollution. And so we're most of the like fertilizer and yard fertilizer runoff, like all of that is kind of exempt from actions that are regulated by the Clean Water Act. And so there could be policy recommendations and management strategies that go towards helping reduce these impacts. Chris, I don't want to cut you off. I think you also have valuable things to say, and I talk a lot, so.
No, I think that's perfect. Yeah, I think the other thing in thinking about this is lots of times recommendations are given, but potentially they're on a voluntary basis because it's tougher to put regulations in place. And I think that's one of the challenges we need to think a little more about is, whether or not, we can model all kinds of best management practices based on like everyone 100% volunteers to do everything possible. And we can see nutrient loading reductions would translate to mitigated algal blooms, things like that. But whether or not that happens in reality is kind of the challenge, right? So I think that's the thing where it becomes really complex is thinking about developing these recommendations and then trying to develop, whether or not it is regulatory or there is a more effective way to do it on a voluntary basis. I think in the past, that's where I've seen lots of programs fall short is they're really good recommendations, but sometimes there's not an incentive or there's not teeth behind it to really make it an effective practice.
Yeah, and I guess kind of to wrap up this Q&A, a little bit of a local interest question and open again to anybody. Jan asks, how does the Park Point area fit into this picture?
I think that one's for you, Sam.
Well, as the least local person on the panel, I don't know if Kate wants to weigh in. I don't know if she's paying attention, but I don't know where Park Point is.
Can you repeat the question? I'm listening, but I wasn't listening close enough to be able to answer the question directly.
Yeah, I guess the general gist of the question is how does the Park Point area fit into the picture of harmful algal blooms or algal blooms in general?
Yeah, that's an interesting question. So in Lake Superior, I started working on this during my PhD, and we started seeing blooms originally up near the Apostle Islands region. And so we were originally thinking kind of like this was like basically like a bloom hotspot near the Apostle Islands sea caves. One of the reasons that might have been was just like part of when we saw those observations where like park rangers were out kayaking and stuff, and so they were seeing this happening. So there certainly could be a component of this that's just like a general awareness. But it does seem like they're starting to like kind of crop up more and more. I would say that in the lake proper, the Park Point, Wisconsin Beach or Wisconsin Point Beach area tend to be less prevalent sites for blooms in Lake Superior proper. But there have been a lot of blooms in the St. Louis River estuary, which obviously, exchanges water frequently with those areas, Minnesota Point and Wisconsin Point. So I think that we sometimes see some maybe like diluted blooms from the estuary, but it's still really, I think, an open question of like, did these cells originate from the lake or did they originate from the estuary? Or, kind of essentially what is the like hydrodynamic history of like where the cells were and how they're growing and things like that. So I'm sorry I can't give you like kind of a clear answer. The exciting thing from the research standpoint is that this is all very new and tricky to understand and like an exciting area of research, but the tough part is that we still are very much in kind of the beginnings of understanding what the heck is going on in Lake Superior, and understanding kind of like the role of inflowing waters in blooms.
Great. Thanks, Kate. And Julie with the buzzer beater here. For anyone, is anyone planning any monitoring of Lake Superior in Minnesota side of things?
Yeah, I can jump in on that one. So kind of in Minnesota at this point, it's kind of on a voluntary basis. This is something I've been talking with organizations about for probably 4 years now about the need to start looking at this. So Minnesota does have a really great beach monitoring program run by Minnesota Department of Health. I know Minnesota Pollution Control Agency is doing really great work as well. But right now, like the beach monitoring program only looks at E. coli and kind of bacterial pathogens and things like that from that perspective for health reasons on whether or not a beach should stay a beach should stay open. But we've been talking for quite some time now about trying to at least integrate some sampling for cyanotoxins into that routine monitoring, because I think that's really what's needed. But again, this comes to the complexity of, who pays for it then? Which organization is going to be the champion of routinely supporting this financially? I think we got the research infrastructure in place. We got the monitoring infrastructure in place. We got the reporting systems in place. Now we just need the money to do the work. And I think that's one of the challenges is right now everyone has such a limited budget. How do you focus those dollars and allocate them? And so I think this is just going to have to be an ongoing conversation at this point.
Yeah, and I think with that, we'll put an end to the Q&A. And thank you so much, Leanne, Sam, and Chris for joining us, and Kate for stepping in there with that question. And just want to thank everybody that showed up and listened to the webinar. Thank you so much for spending your time with us. And I think I'll pass it back to Julie for any closing thoughts.
That's it. Just a real great thank you. This is really good information today, and I'm actually looking forward to checking out the webinar too. Jesse wasn't able to join us the last minute, but there's great information, and hopefully we can add some more monitoring sites in Minnesota and the Lake Superior nearshore area. So thanks, everyone. Appreciate all of your participation.
Thanks for organizing. This is great. Bye.
Thank you. Bye-bye.